Welcome to part 3 of the pVACview demonstration. In this video, we will use pVACview to explore our data. The first thing you see upon switching to the Explore tab is the main aggregate report table. By hovering over each individual column header, you will be able to see a short description for the respective column. For columns such as the predicted IC50 binding for mutant peptides, we have overlaid a color scheme with different shades of green representing binding under 500 nanomolar, shades of yellow and orange representing binding between 500 and 1000 nanomolar, and red representing those above 1000. Similarly, the cutoffs for the percentile columns are between 0.5 and 2%. All candidates are given a tiering based on whether they pass certain criteria. The specifics of this tiering metric are documented in our help docs. For each candidate, we use red boxes to highlight which criteria the candidate is failing. Candidates are automatically sorted with passing peptides at the beginning of the table and candidates with no expression at the end. Now let's pick out a candidate to investigate further. Clicking the respective Investigate button located at the end of each row will allow the app to search and load transcript and peptide level data for the variant of interest. Scrolling down, we can see more information presented in the box below. On the right, there is some additional variant and gene information, such as DNA and RNA VAF, gene expression, and genomic information. We also provide a link to the corresponding Open Cravat variant report. This report contains additional information such as clinical relevance, functional studies, and related literature if any is found. On the left, we have the transcript sets covering this variant that produce strong binding peptide candidates. These transcripts are grouped such that if multiple produce the same set of peptides, then they belong to the same set. Here we have two transcript sets, with one containing 14 transcripts and the other with just one. The first and second sets both produce 3 peptides. Total expression is calculated by adding the transcript expression values in each set. When the reference proteome similarly feature was enabled during the original pVACseq run, the reference match tab will show the best peptide, the larger peptide sequence around the best peptide that was queried for, and it will list the hits that were found in the reference proteome for that query sequence. For this candidate, there is no reference matches. However, if we investigate this candidate, we see a peptide that matches 7 out of 9 amino acids of the best peptide. The possibility of this peptide binding to the wrong gene is concerning and could result in this candidate being rejected. If you choose to upload additional data, the third tab on the left will show the median IC50 prediction regarding peptides containing this variant. In this case, I uploaded additional Class 2 data, and the results are shown accordingly. For a summary of this information, please see the Tutorials tab. For further documentation, please refer to the pVAC Tools GitHub page and the Read the Docs website.